Opportunity. Well, we are perfectly in time, and I invite my friend Professor Emidio Scarpellini, researcher to University of Leuven in Belgium, to present his speech, Gut Microbiota and Hepatocellular Carcinoma Development. Please, Emidio.
Hi to everybody. Just a second. Yeah, here I am. So first of all, thank to Ludovico for inviting me in, to all the editorial board for this exciting opportunity. Today we'll try to speak about the gut microbiota and hepatocellular carcinoma natural history and development in particular. What is gut microbiota? We can say that it's the largest, the biggest symbiotic ecosystem within our body. It's about 2 kilos weight, and is not only composed by bacteria, but also from other microorganisms like archaea, protozoa, yeasts, and viruses, most of all. It's about several trillion of microbes, and the main bacterial components of our microbiota are Firmicutes, Bacteroidetes, Proteobacteria, Actinobacteria, Verrucomicrobia, Fusobacteria, and Cyanobacteria. Our human health is affected from a multidimensional point of view by these bacterial phyla, and as you can see here, these are the main one divided by gram-negative and gram-positive. What are the functions? We describe it briefly in here as my colleagues did partly before. The functions of gut microbiota in our body, as you can see, they contribute to the absorption of micro and macro nutrients like proteins, fibers, fat digestion, and they participate in the biosynthesis of neurotransmitters, BCAA, short-chain fatty acids, triglycerides and fatty acids, and vitamins as well, neurotransmitters and hormones as well. So they affect the functioning of our brain, of our circulatory systems, the adipose tissue in a bidirectional way, kidneys, and so on. So this is a little bit of taxonomy, just to remind that about the bacterial components of our gut microbiota. We can recognize phylum, family, genus, and species. And here, among the main phylum, we remind and we record Firmicutes, Bacteroidetes, and Actinobacteria. And this is a nice picture where we need legend just to understand what's going on. So from neonate infant until our old people, we can see that the amount of represented phyla of bacteria, they change dynamically. So the beta diversity, that it's the difference of microbial composition between subjects, the inter-individual variability, is going down when we become older and older. Meanwhile, the bacterial diversity should increase, so the alpha diversity, but towards several ways of alteration of the composition of our microbiota. And why it happens? As my colleagues already mentioned, there are age-specific factors, genetic factors, and Professor Bocuto in detail depicted this very complex relationship, let's say, between microbiota and the host and environment and so on. And we see that here we have different factors that can be comorbidities like obesity, type 2 diabetes, that affect the composition of our microbiota, not excluding the aging. So far, how does it work, our gut microbiota? And it helps us to understand how it interacts with the liver. So in our gut, there is fermentation of carbohydrates, fibers. So they access through the blood stream to the liver. They become metabolized. But this happens not only for nutrients of any origin, but also for toxic substances. So also for our bile acids. So the 1-2% of enterocholic recirculation of our bile acids is a small amount in consideration of all the amount of bile acids that we produce circulates not only in the gut. But this small amount condition gut microbiota composition and most of the metabolic processes happening within the liver and in the rest of the body. And of course, our gut microbiota contributes with antigens presentation, the gut and the systemic immune system education to the development, maintainment of immunity, tolerance, and in disease presentation. So what happens when our gut, liver, let's say immune axis works? Well, what we say. So here there is our normal gut microbiota or aerobiosis condition. Tight junction regulate intestinal permeability to antigens, to toxic substances, but mainly to antigens. So a few of them will be presented to the immune system through the portal vein circulation into the liver, Kupffer cells, stellate cells, TLR4. When there is gut microbiota dysbiosis, there is an increased or impaired, let's say, intestinal permeability. Pumps, dumps, they pass through blood circulation, portal circulation into the liver. There is an hyper and not physiological activation of our immune system. There is inflammation, fat deposition, inflammation all together. They may lead to cirrhosis and to HCC development. We can jump from NASH to HCC, like my colleagues previously reminded. And in detail, what happens? That these dysbiosis affect bile acids metabolism, ethanol production with hyperproduction and altered metabolism, choline providing to our host intestinal permeability, so perpetuating this vicious circle responsible for our liver disease. And this is an example, a very nice figure from this very nice review, very updated by XU, that shows how every liver disease has a proper dysbiosis. So alcoholic fatty liver disease, NAFLD, NASH, and MAFLD, the bigger entity that we are coming to get in touch and playing with, the hepatocellular carcinoma, and also the autoimmune hepatic disease, including the cholangiopathies. And this is what we did with Ludovico very recently. We had a chance to publish this work, trying to study ex vivo what happens to gut microbiota dysbiosis in a cohort of cirrhotic and non-cirrhotic patients from our center here in Marche. And what we saw, analyzing with the anthropometric and patient characteristic, that the two groups of cirrhotic and non-cirrhotic, they were quite similar. There was a very mild difference about autoimmune or cholangiopathys representation, but was not statistically significant according to the sample size. What it was interesting and quite overlapping and so confirming literature data, there was a significant prevalence of small intestinal bacterial overgrowth in both cirrhotics and non-cirrhotic patients versus healthy volunteers. And this data was more significant for hepatic encephalopathic cirrhotic patients. What we saw dosing endotoxin levels is that there was as well a significant difference of cirrhotic and non-cirrhotic patients versus healthy volunteers. But this difference was even more significant according to the gravity, so the stage of gravity of liver disease, according to child path status. Looking, and this is the most important slide, at the eventual correlation or relationship between small intestinal bacterial overgrowth and blood concentration of endotoxin, we saw the very interesting result, that is this, that SIBO absence is not compulsory for absence of higher levels of endotoxin. So probably the small intestinal bacterial overgrowth describe a very upper GI tract gut dysbiosis that does not describe the passage through an altered intestinal permeability of endotoxin, so lipopolysaccharide, that is a pump. So these are the study linking in animal models of liver disease, so cirrhotic and non-cirrhotic, with or without hepatocellular carcinoma, gut dysbiosis, with occurrence of liver disease, occurrence of HCC. This is the rest of the slide, and here we can clearly see that there is a correlation between dysbiosis. The dysbiosis can be different according to the different model of liver disease use, either inflammatory, either diet linked with the high-fat diet, or toxic model of liver disease. What about human data? Are they in? Yes, there are several. So this is a solid evidence as well. And we see that even here, with or without HCC development, with or without liver cirrhosis, there is a consolidated literature data showing that there is an heterogeneous gut dysbiosis according to the presence of liver disease, to its stage, and or to the presence or absence of hepatocellular carcinoma. What about dysbiosis and liver regeneration? Is there a relationship? Yes, it's there. So we see that the regeneration of the liver is affected by several cytokines, hormonal, not hormonal agents. And then there is a termination phase, mainly affected from TGF-beta. What happens in an animal model where we operate a partial hepatectomy? that there is a variation, a very early variation in terms of days or hours of our microbiota. So we can imagine what happens, the shift that happens, especially in the liver cirrhosis patients that are developing or not developing hepatocellular carcinoma. And this is one of the most nice study showing in an inflammatory model of liver disease leading to hepatocellular carcinoma. What happens? So in terms of dysbiosis, we see that there is during liver inflammation, so we can imagine an ASH patient, for example, a microbial gut invasion of the intestinal mucosa, so pumps, they pass the altered intestinal permeability, and we arrive until liver cirrhosis. Here there is an higher lipopolysaccharide biosynthesis. And it's interesting that our HCC get nourishment from not only from carbohydrates, but starts the switch from proteins. So also the metabolome within our liver and our intestine changes. What about systemic inflammatory response? Here you can see lipopolysaccharide levels going up, and you see our systemic immune response that beforehand was very intense on the GI tract and also in the liver going down. And it increases our inflammatory response within the liver. So the tolerance is going down. So the surveillance towards possible hepatic cells that under this push can get mutation and become hepatocellular carcinoma. So that's what happens inside the liver, like we were saying. Oh, the inflammatory response works. No, and now it doesn't work anymore. So our T8 cytotoxic lymphocyte, they go down. Then they don't get surveillance towards our hepatocellular potential cells over proliferation. But this is an active process. In this very, very nice model of inflammatory liver disease, we can see what? That deleting the NLRP6 NFKB control gene in these mice. So this is the group. We have a significant, a dramatic shift at 13 weeks time point of gut microbiota. And in particular, Akkermansia mucinifila goes down in terms of abundance. Intestinal permeability in this kind of modified mice towards the control model increase. And what happens if the intestinal permeability increase? You see these deleted mice, the tumor nodule size significantly increase. Further, you see that according to the presence or absence of NLRP6 deletion, it increases the amount of the mesenchymal monocytic stem cell suppressor that go to suppress our cytotoxic T cell CD8, and you can see clearly. And you see the inverse correlation. The more our mesenchymal cell increase within the liver, the lower it becomes the concentration of the tolerance cell, let's say, that maintain surveillance towards the tumor cells. What happens if we give antibiotics cocktail to these mice? We see that significantly these suppressor cells go down, and we see that in case of these NEMO modified mice, this is not true anymore. What happens if we operate a fecal microbiota transplantation of NLRP6 deleted mice to the, let's say, control group, the NEMO delta EPA group of mice, that it is reverted the concentration of suppressor cell, and the cytotoxic T8 cell, they go down as well here. But very nicely, that what it happens as well in terms of abundance of single phyla of bacteria. So if, as we operate the fecal microbiota transplantation, the quote of suppressing cells or CD8 cytotoxic cells increase, and the abundance of Akkermansia mucinifila goes down. What in terms of treatment response? So are there bad bugs for response to anti-androgenetic and checkpoint inhibitors in HCC evidences? Yes, there are several. So these dysbiosis affect the response to several chemotherapy in treatment of HCC. At this point, most of us know that in comparison with cholangiocarcinoma, in cholangiocarcinoma, we know about 40% of mutation. So we can more easily use checkpoint inhibitors, drugs, just to prolong survival of these patients, to improve their quality of life. And we hope to reduce volume and to improve expectancy of life for this patient, at least in the next 5 to 10 years. In terms of knowledge of HCC, we don't know almost anything about the mutation. We know about, in comparison with cholangiocarcinoma, 2.5% of mutation. So we will need a biomarker that allow us to make an early cancer diagnosis and to predict treatment response, especially with the newer checkpoint inhibitors that have been used in the last years for HCC treatment. Are there evidences? This is a very nice Chinese study where we see that we start to see the mutation of different Escherichia coli, in particular strain, with this marker here below, and we can get a prediction of early hepatocellular carcinoma presence, so diagnosis, and response to chemotherapy, because we know that alpha-fetoprotein and other biomarkers derived from alpha-fetoprotein, very often, let's say in 40–50% of cases, they don't allow to get even a suspect diagnosis of early HCC. What about gut microbiota and other biomarkers and response to treatment? Do we have other data? Yeah, that's a very nice study from my ex-university, so Catholic University of Sacred Heart, Gemelli Hospital, and that's the friend Francesca Ponziani. They performed a small study, but very nice, where mechanistically they saw gut dysbiosis improving and fecal calprotectin concentration decreasing in those patients getting a good response and tumor diameter reduction after immunocheckpoint inhibitors usage. And what is the mechanism? Well, the mechanism through which modulation of gut microbiota with antibiotics, probiotics, let's say also postbiotics, we are in the age, and metabolomics and so on, and the omics, as Professor Bocuto magistrally said, are different, so we can act on bile acid metabolism, on immune response, short-chain fatty acids synthesis, cytokines synthesis, and factors that regulate hepatocytes, mitosis, and apoptosis. Is a gut microbiota target for HCC modulation? Are there evidences? Well, yes. There are several evidences uses both probiotics at a very high concentration, antibiotics cocktails that are showing some data, very interesting data, at least on a reversal of dysbiosis and improvement of HCC behavior. What about men or human studies? Where there are still a few, and there is some first hint on the significant association between gut dysbiosis shift towards eubiosis and faster or better reduction of hepatocellular carcinoma dimensions, improvement of patient and liver function. So what are our concluding remarks to make a summary? That we have a different gut microbiota composition, not necessarily we can speak about dysbiosis in every hepatic disease at any stage. The dysbiosis is of course involved in pathophysiology of hepatocellular carcinoma. We focus on the gut-liver immune axis. The dysbiosis has been confirmed to be present in different animal models and human studies about HCC development. Gut dysbiosis, and this data is crucial for me, is responsible for the immune response switch within the liver, both for tumor initiation and progression. Certain gut dysbiosis subset hallmark are associated with certain HCC responses to both anti-androgenetic and checkpoint inhibitors agents. Gut microbiota can be used as a marker, perhaps, for early HCC diagnosis and treatment response prediction. Yes, we have the first promising data. Finally, the gut microbiota modulation show promising first results in humans and also according to animal data as being an add-on treatment together with the chemotherapy. And the last one, the most important slide for me, I thank you for your attention.
Thank you, Emidio, for your excellent presentation. Maybe at the end of this session, we can speak about other interesting points and to reply to different aspects. Yeah. I continue.